Hello dear students, in today's lecture we will be talking about the pathogen involved in nosocomial infection part 2. In a previous lecture, if you remember, we have talked about two important pathogens, Staphylococcus aureus and Klebsiella pneumonia, how they are resistant. In today's lecture, we will talk about two other important bacteria. The third one is Acinetobacter baumannii. It's a gram-negative bacteria. It means it has a thin cell wall and an outer membrane, so it stains pink in gram staining. But why is it important? Because it is mainly found in hospital, and our lecture is about those pathogens that are involved in nosocomial infection. So they cause hospital-acquired infections, especially dangerous for patients in ICU and people with weak immune system, or we can say immunocompromised system. Healthy people usually fight it off, but weak patients cannot. Why is it dangerous? Because this bacterium is important. It has three major abilities: the pathogenicity, causing disease; it can infect lung, blood, wound, and urine. Antibiotic resistance. It is resistant to many antibiotics, so it is very hard to treat because many antibiotics do not work on it. Survival in harsh environment. It can live on hospital surfaces, medical equipments. It survives dry conditions, which is unusual for bacteria. In virulence factor, we have the biofilm formation. A biofilm is like a protective slime layer that bacteria make to cover themselves. Think of it as a shield that sticks to surfaces. Where does Acinetobacter baumannii form biofilms? It is commonly forms biofilm on medical devices, especially urinary catheters, the tube used to drain urine, ventilator, which used for patients who need breathing support. The image you see in the figure in this slide, it shows catheters connected to urine bags. These devices stay inside the body for a long period, giving bacteria the perfect place to stick and grow. Why this is important? Because when Acinetobacter baumannii forms a biofilm, antibiotic cannot easily reach the bacteria because the biofilm acts like a helmet. It's a shield, as we said before. It protects the bacteria. So the bacteria become very hard to remove. Even cleaning and disinfecting the device may not be enough. It allows the bacteria to survive for a long period in hospital. This is why Acinetobacter is known for causing persistent hospital infections. Imagine bacteria building a sticky home on the catheter. Inside that home, they hide from antibiotics, the patient's immune system, and cleaning agents. The bacteria also reduce metabolic activity. Bacteria inside biofilms grow very slowly or enter a resting state. Most antibiotics target actively growing cells. So when the bacteria slow down their activity, the antibiotics become less effective. And also biofilm protects bacteria from phagocytosis and other host immune response. They could be associated with chronic and recurrent infections as they are difficult to eradicate completely. After that we have outer membrane protein. This protein helps the bacteria survive in harsh conditions, such as those found on surfaces and medical equipment. The outer membrane, the bacteria, is a protective shield, act as guard at the gate. They help block harmful things like disinfectant and antibiotic. They make it hard for medicine to enter the bacteria and help Acinetobacter pneumoniae to survive or live in hospital beds, medical equipment, surfaces. Also, your body tries to kill bacteria, but outer membrane proteins help the bacteria hide or resist immune attack. The structure and function of outer membrane proteins. The outer layer of bacteria contains lipopolysaccharide, act like a strong outer shield, and then phospholipids form the basic membrane structure. Outer membrane protein, special function protein. This outer membrane is like a wall made of bricks, plus special door and tools. Porins as channels. Some outer membrane protein act as porins, tiny holes or channels. They allow nutrients, small molecules, and ions to go in and out of the bacteria. Then the adhesion and invasion. Some outer membrane protein help the bacteria to stick to human cells, attack the tissue, and the bacteria can grow and spread. The fourth one is immune evasion, escaping the body's defense. When our immune system tries to destroy the bacteria, outer membrane protein help the Acinetobacter to hide from immune cells, avoid being recognized, resist killing mechanism. If we look at this figure, we are going to see that the bacteria is like a layered sandwich. The first one, cystole, inside of the cell. This is the innermost part. It contains DNA, ribosome, and everything needed for life. The inner membrane is a thin membrane surrounding the cytosol, and control what goes in and out of the cell. Then we have the periplasmic space. This space between inner and outer membrane. It contains peptidoglycan, a thin cell wall layer, enzyme, and proteins. This space helps us structure, nutrient process, and defense. The peptidoglycan layer is strong, but it's very thin. Give the bacteria shape and support. And then we have the outer membrane. This is what makes gram-negative bacteria special. It contains lipopolysaccharide found on the outer surface, act as protective shield, can trigger immune reactions. The porins in the outer membrane protein, as we said before in the previous slide, shows us an orange tube in our diagram. Small channels that allows nutrient in, waste out. They can block antibiotics. And other outer membrane proteins help in attachment, stability, and immune evasion. Lipoprotein, the connect layers, it connects outer membrane to peptidoglycan. If we look at the right side, we see that the entire bacterial cell, it has a capsule, pili, flagella, nucleus. This is a multi-layer system: strong protection, controlled entry, survival and advantage, and infection ability. Antibiotic resistance in Acinetobacter baumannii. It's a multi-drug resistant. It means that the bacteria can survive many different antibiotics. Resistant to strong antibiotic, well, such as carbapenems and colistin. Carbapenems, which last strong option, and colistin is last resort. Even the strongest medicine may not work anymore. Acinetobacter baumannii can share resistance gene with other bacteria. It uses plasmid, which are small circular DNA, and integron, gene collector. Bacteria passing survival instructions to each other. And then bacteria quickly become resistant to many drugs. Infections become harder to treat, longer lasting, and more dangerous. Because in healthcare setting, many antibiotics are used, bacteria spread easily, spread fast, cause serious infection, is difficult to control. In this slide you will see that how the bacteria defeats a strong antibiotic. It's resistant to carbapenems in they are very powerful antibiotic and used when other drug fails. It's a last strong weapon doctors use. Acinetobacter baumannii can escape the effect of the drugs. How? The main mechanism, they are enzymes, special protein made by the bacteria. It breaks down carbapenem antibiotics. They cut and destroy the antibiotic molecules. This process is called hydrolysis. So the antibiotic becomes inactive or useless. The carbapenem, it cuts the key before it reaches the lock. Let's say it looks like a key and a lock. Antibiotic is the key and bacteria is a lock. These are specific carbapenemases. They are very common in Acinetobacter baumannii. They are one of the main reasons this bacteria is dangerous. And then we have purine loss and efflux pumps. What are purines? As we said before, they are tiny channels which allow substances to enter the cell. It means these purines allow substances including antibiotics to enter the cell. So what does the bacteria do about this? It can reduce or lose these purines so that there are fewer doors and less antibiotic enters. Then you have the efflux pump. They are a special protein that acts as pumps. It increases, upregulates these pumps, activate pushes antibiotic out of the cell. Then antibiotic when enter the cell, efflux pump is a security guard. It throws the antibiotic out immediately. So antibiotic cannot stay inside and drug level becomes too low to work. If we look at this figure, we see that purines are special outer membrane proteins. As we said before, they found in gram-negative bacteria. So purines let small substances pass in and out of the bacteria. Nutrients, ions, small molecules, even some antibiotics. This happens by passive diffusion. No energy needed. Let's say that these purines are small open windows in a house. Good thing that food and air come in, waste goes out. But sometimes harmful things like antibiotics can also enter. So why are purines important in antibiotic resistance? Purines play a key role in whether antibiotic can enter the bacteria or not. If purines are reduced or changed, fewer windows will be open, so antibiotic cannot enter properly. So what efflux pumps are? As you can see in the figure, they act as a pump. They pump the harmful substances out of the cell. Efflux pumps can push out antibiotic, disinfectant, and other toxic substances. So how do they work? When antibiotic enters the bacteria, the pump actively pushes it back out. This process uses energy, and so it's not active, not passive, like the purines. Now we have colistin resistance in Acinetobacter baumannii. Colistin is a last resort antibiotic. What does last resort mean? It means when all the other antibiotics, when they don't work, they use colistin. It is our last option, which targets the outer membrane of gram-negative bacteria. Specifically, it binds to a part called lipid A. What is lipid A? Is a part of the outer membrane. It helps maintain the structure of the bacteria. So the bacteria added chemical groups, positive and cationic groups. For example, 4-amino-4-deoxy L-arabinose. So colcitin, it looks like a magnet. And lipid A, metal surface. Magnet which sticks to the lipid A, then bacteria dies. But bacteria change the surface. The magnet cannot stick anymore. That is why colcitin cannot bind properly. Outer membrane is not damaged and antibiotics become ineffective. There are also other ways for Acinetobacter baumannii to resist colistin. We have the acquired resistance via plasmid. The bacteria can get resistance from other bacteria. How does it happen? It's raw. Plasmid, as we said in the previous slide, what were plasmid? They were small DNA circles, integron, and which are gene collectors. Think of bacteria like a student. When one student has answer, they share it with others. So many bacteria becomes resistant. Resistance spread very fast. Even previously sensitive bacteria become resistant. The second one, the efflux pumps to colistin as well. It can pushes antibiotic out, like it was in the previous. So drug cannot stay inside and it becomes less effective. The bacteria can also modify its cell surface, change membrane structure. This makes it harder for colistin to bind and damage the membrane. And then we have another important bacteria, which is very dangerous. It's called Pseudomonas aeruginosa. They are opportunistic bacterium, especially in hospitals and immunocompromised patients. Their virulence factors uses to infect and damage the body. It has a type 3 secretion system, which is a special system that injects toxins directly into human cells. And then we have the production of biofilm. Also like the Acinetobacter baumannii, it form biofilms, which is a sticky substances produced by bacteria. Think of it like a slime or glue. Bacteria stick together, attached to surfaces like lung, catheter, etc. So, biofilm is a protective layer of bacteria. Bacteria are very hard to kill. Antibiotics don't work well. Then we have the quorum sensing. Bacterial communication is a system where bacteria talk to each other using chemical signals. They control toxin production, biofilm formation, and virulence gene. Type 3 secretion system in Pseudomonas aeruginosa, as we said before, that it injects into the human cells. It injects toxin into the human cells. It's a needle-like structure, which used by the bacteria to inject harmful toxins. Once inside the host cells, they disrupt cell signaling, so the cell cannot function normally. Block immune response, the body cannot fight the bacteria effectively, and also lead to infections and diseases. It's a major virulence factor, help the bacteria invade the body, survive immune attack, and cause severe infections. That is why type 3 secretion system is very important. Where is it especially important? In patients with cystic fibrosis. Cystic fibrosis, which is a genetic disease. Lungs are already being weak and bacteria easily infect. Also in burn wounds, damaged skin, easy entry for bacteria. Cystic fibrosis, as I said before, is a genetic disorder that primarily affects the lung, pancreas, and other organs. It's due to the mutation of the cystic fibrosis transmembrane conductance regulator gene, which leads to the production of sticky mucosa, which cause respiratory and digestive problems. Clinical manifestation. This likely refers to how Pseudomonas aeruginosa infection appears in patients. Now, the bacterium cannot infect many parts of the body, especially in weak or hospitalized patients. The respiratory infections, it's especially in cystic fibrosis patient, ventilated patient. The symptoms are cough, thick mucus, difficult breathing, and pneumonia. The second one is skin and burn infection. In infects burn, which cause greenish pus, bad smells, and delay healing. Then we have the ear infections, cause swimmer's ear pain, discharge, and swelling, and also eyes infection, especially in contact lens users. Symptoms would be like redness, pain, and vision problem. Also, bloodstream infection, which is very serious, and the symptoms are fever, low blood pressure, shock. Urinary tract infections, common in catheterized patients. The pain during urination, fever and cloudy urine. And also digestive system problems, which is pancreatic insufficiency. The pancreas cannot release digestive enzymes anymore. So it would be poor digestion of fats and poor absorption of vitamin A, D, E, and K. This is called malabsorption. The second one is meconium illness, which is in newborns. Sick stool, block the intestine, and intestinal obstruction at birth. The liver disease, this mucosa blocks bile ducts, liver damage and scarring. Salty skins, loss of too much sodium in sweet, and then infertility is also common in this case. Absence of vas deferens, tube-carrying sperms. Males are often infertile. Now if we look at this diagram, it's a biofilm life cycle. How biofilm is being formed. Think of biofilm formation like bacteria building a protective community. The first one, initial attachment. Single bacteria land on a surface. Example, lung, tissue, catheter, wound, or other medical equipments. When the bacteria arrives and start exploring, bacteria stick to the surface and start sticking to each other. They begin form a small group. Microcolony formation. Bacteria multiply and form small clusters. Then maturation. Also, bacteria produce the alginate is look like the biofilm, which becomes thicker, stronger and well organized. So it's a fully developed bacteria. Then we have the dispersal. Some bacteria leave the biofilm and travel to new places. Why is this important? Because biofilm make bacteria resistant to antibiotic, protect from immune system, and cause chronic infections, especially dangerous in cystic fibrosis, as we said before.